So this is actually how I got into this kind of field. My day job is epilepsy surgery, but I was reading that somebody said that puberty blockers were completely irreversible, and from a neuropsychological perspective, that seemed really surprising from everything we know about brain development. So I was really excited. I thought this is fascinating. So I went to look at the literature that shows us that they're reversible, and this is kind of a result of that exploration, really. So just where I'm going to be going over the next 20 minutes or so, I'm really going to cover why cognition is an important area to assess if you're going to put somebody on to puberty blockers. So I'm going to give you a very brief overview of brain development. You've already had a bit of brain stuff this morning. You've got some more coming at you now. And then I'm going to just introduce you to the concept of critical windows of opportunity when it comes to brain development, because these are really important windows where the brain is developing, and we need to be very careful of the input that's going into somebody when that's happening. I'm then going to talk about the animal studies that we know that have looked at the impact of puberty blockers in animals, and then I'm going to talk about the studies in people. So brain development, understandably, is incredibly complex. It's not just that you're born with a brain and it gets bigger and bigger. There are all sorts of phases of brain development. So the adult brain will have roughly 100 trillion synaptic connections in the adult brains, but that doesn't develop linear. It's not in a linear fashion. So if we're thinking about brain development, you get progressive events where you've got lots and lots of connections being made between the neurons, and then you also get regressive events where those connections are pruned back. And the progressive events, all the connections, are just as important as the regressive events when you're pruning back the connections that you've made that you don't actually need. So you get cell death. Some of the cells actually die. They are programmed to die. They are supposed to die. It's part of the development. You get synapse elimination, so you can get all of these connections, and then you don't need that network anymore as you get older. And you also get axon pruning. So the most important thing to kind of realise about brain development is that it doesn't just get bigger and just more connected. It's connecting, and then it's looking at those connections and getting rid of the ones that they don't need, and then getting new connections. And that happens in a very set way throughout development. And what we know is that there are developmental windows of opportunity. So the brain is pre-programmed. When we are born, we are pre-programmed different bits of the brain to connect up at different times. And during these windows of opportunity, different aspects of behaviour and cognition are kind of prioritised. So when you are first born, the thing that's really important is that you learn how to see. So the vision bits get connected up, and then you have to learn how to control movement. So the movement bits get connected up, and then a little bit later on, you've got the language bits that are really focused on, and those bits are being connected up. So all of that is happening at different stages of development. And what we know from some pretty grim animal experiments, actually, is if you deprive the brain of any input during these critical windows of opportunity, the brain will move on, and that whatever it was that was supposed to be developing doesn't develop properly. So we know that when they've sewn cat's eyes together and they couldn't see for a certain period of time, and then they let them see, if that window of opportunity passed, they never actually learned to see properly. So you don't catch up. The brain has moved on. It's moved on to something different. And we know this in adults from the development of language. So if you think about language, we develop language when we're 2 and 3. We start to learn to talk. If you try and learn a second language when you're 2, 3, 4, you'll learn it really well. And actually, you can learn it without an accent. You can learn it completely accent-free. The window for acquiring language begins to close as you get into your teenage years. If you try and learn a second language in adulthood, you will never learn it without an accent. That window is closed. You will learn it. There will be bits you can learn, but actually that window of opportunity to acquire language has closed. So the critical windows of opportunity throughout development are really important when it comes to thinking about how the brain develops different functions. And it just so happens that adolescence is another critical window of opportunity and of neurodevelopment. This is when lots and lots of pruning occurs. So it's a regressive stage in terms of all of the neurons that are getting connected, a regressive stage of neurodevelopment. So if you look at the diagram there, you've got at birth, you've got very few connections. By the time you're 7, you've got loads and loads and loads of connections. The brain is just connecting all the time because it doesn't know what's important. So everything that it encounters, it's like, this might be important, make that connection. By the time you get to adolescence, that's when you start to think, actually, that's not important. Maybe Santa Claus doesn't exist. Maybe I don't need that. Maybe I don't need to be leaving my teeth under the pillow for the tooth fairy. Maybe other connections are more important. I need to maybe brush my teeth every day because that seems to have a good impression on the people around me. So you're learning different things as you are developing. You're pruning back and you're strengthening new connections. And adolescence is a critical window of opportunity for that. And in particular, we're really looking at the frontal circuit. So that's the bits at the front of the brain. And that's everything to do with executive function and social cognition. And it's why adolescents go through such a difficult time in puberty, because they're negotiating all of that, and all of that is developing. So that pruning that happens in adolescence is really, really important. And the key thing about that pruning is that it's not dependent on age. It's dependent on pubertal stage, so it's actually hormonally driven. It's not an age thing, it's driven by puberty. The other thing about the kind of developmental stage during adolescence is that there are sex differences. So I said this morning that there aren't any sort of differentiating sex differences when it comes to brain volume and brain structure and things like that. There are very significant differences when it comes to development, neurodevelopment, because the brain is responding to the sex receptors within the brain are responding to different inputs. So they will be responding to testosterone, or they'll be responding to estrogen. These sex receptors are in different parts of the brain. They've got different densities in different parts of the brain. We're particularly interested, as I said, in the frontal regions of the brain. We're interested in the hippocampus, the amygdala. So that's memory, that's learning, that's emotional control, and all of the executive functions that are associated with frontal lobe structures. So what we see in terms of adolescent brain development during puberty is that structures with a high density of sex steroid receptors, so that's the prefrontal cortex, the amygdala, these are the ones that really start to connect up. So you get lots and lots of connections forming between these structures. And that's driven by the different sex hormones that are produced. So if we're looking at the kind of overall maturation that we see, I mean, anyone who's got a teenager or who's been a teenager will kind of know this. This is what you're developing as you move from being a child to an adult. You're looking at the executive function. So you're looking at working memory, the ability to hold something in your mind and manipulate it at the same time, self-monitoring, planning, prioritization, task initiation, organisation, impulse control. So this is why adolescents can be really, really impulsive, because those regions are still getting connected up. And emotional regulation, they can really just lose their temper at nothing, because again, those connections are being made. Flexible thinking and the ability to appreciate nuance. You need to go through adolescence, you need to go through puberty to really develop these things, the appreciation of nuance, moving away from black and white thinking, and the idea that things may be nuanced is something that is really critical in terms of adolescence. And also social cognition, just where you fit in the world. So all of these things we know, adolescent psychologists, psychiatrists, you will know much more about this. You will see this in your everyday practice. This is what people are struggling with. So adolescence is a second window of opportunity from a neurodevelopmental perspective. So I know all this, and that's why I was really, really surprised to read that you can just pause puberty and then pick up 2 years later or 3 years later, and nothing will happen. Because everything we know about the other windows of opportunity tells us that that's not the case. So if that was the case, that would be amazing. So what's the evidence for that? Let's have a look at that. So just before we go on to that, so just to recap, brain development isn't linear. It follows a predetermined path. Puberty triggers a complex cascade of events in the brain that are critical in that process. Particularly the development of executive function, social cognition, memory, new learning, emotional regulation. So there are very good reasons to be concerned about the impact of any treatment that might interrupt this. Okay, so what's the evidence? Well, so we've got some behavioural studies. There are many, many animals that have been studied. I think there's 17 in all, but most of them that have looked at cognition have been done in sheep. It's the same flock of sheep, actually, that have gone through this, and mice, and also some monkeys. And when they're looking at cognitive kind of markers of cognition in these animals, really they're looking at positive interactions with the environment, so how kind of exploratory are these animals. They look at how much they move about, how much effort they put into trying to get food for themselves. Are they interested in trying new foods? How social are they? And then they also look at how responsive they are to stress. So are they resilient? Do they develop coping mechanisms for stress? And they also look at cognitive tasks, so you can put some of these animals in mazes and see if they can work out the way out of the maze, or work out where to find the food in the maze and things like that. I've just put that up there just so you can see the studies that I pulled out, so that these are the studies that I'm basing my conclusions on that I'm about to tell you. Okay, so if we look at all of these animal studies, really we find that treatment with a puberty blocker has a detrimental impact on learning, and the development of social behaviours and responses to stress. So a detrimental impact on all of those areas of function. And they're sex-specific, so they're different for the male animals to the female animals. In male sheep, and they've only looked at it in male sheep, the impairments in spatial memory that happened with the treatment were not fully reversed when they stopped the treatment and the puberty started, so they're not fully reversed. And significant effects of treatment were also evident on brain structure, and that included volume and functional connectivity and neuronal density. So exactly what we would expect, given what we know about...
That's the one sentence question, I'm sorry. Do all these studies, is follow-up late enough so that catch up if it occurred one at that time?
So there's very little that's been looked at. I think there's one study that's looked at catch up. I don't know whether it's long enough in a sheep. So it was, I think it was 6 months or 9 months or a year later. So I don't know what that means in terms of pubertal catch up, but certainly they hadn't caught up when they did the follow-up. But one of the key things that I'm going to say about this is we do not know, because nobody has looked at this. And that's the kind of, it's a spoiler alert because that's my last slide, but my last slide really is, how can we say that these things are reversible when nobody has bothered to look? And it is an outrage that nobody has bothered to look, given what we know about brain development. But that's me. Okay, so the summary of the animal studies really is that they're fairly consistent. They indicate that the suppression of puberty impacts brain structure and the development of social and cognitive function in mammals. The impacts are quite complex. They're also sex-specific, so we need to be aware of the different ways that the male and female brain develops. There's no evidence that they're reversible. There's no evidence, but there's one study that suggests it's not completely reversible. But let's move on to people, because, animals are one thing, but it's the people that are receiving these medications. We are giving these medications to young people. If we look at the studies that have actually looked at formal assessment of cognitive function, there are very, very few. I found 5 when I was looking in the literature. There were 2 that have looked at cognitive function in groups of people who've undergone precocious puberty, and I found 3 in transgender people. So This is one of the only studies, a group study, that looked at a measure beforehand, so actually got a baseline, and then looked at them after they'd been treated. So they used the Wechsler the Wechsler Intelligence Scales for Children, which are a very good standardised test of IQ. And they looked at 25 girls who had been treated with puberty blockers, and they followed them up 3 years after they'd been on the puberty blockers. The group had an average decline of 7 IQ points over the course of their treatment, but at least one of those, and this was the only one I could get from the table, the way it had been presented, at least one of those people had an IQ drop of 15 points, because the highest score beforehand was 138 and the highest score afterwards was 123. So there were some significant declines in IQ in that particular group of who had precocious puberty. This is a second study that looked at precocious puberty, Norway, again a fairly small group, 15 girls, and they didn't do a baseline, so this is just comparing their IQ with a group of controls, and they had a 7-point IQ difference. They said it wasn't statistically significant, but it was probably underpowered. They also found that the girls, puberty-blocked girls, perform more poorly on the sequencing task. But let's move on to the transgender literature. So there were 3 of these studies. In this particular study, there were 20 trans patients. Some of them were male to female, some of them were female to male. And they compared them to a control group on 4 subtests of intelligence, and they also used the Tower of London test, which is a kind of strategy test. It's an executive function task. So it's a slightly strange paper, because they report some of the tests that they did, but then they don't report the results of those tests. So they did tests of mental rotation and they did tests of face recognition, but we don't know what the results are. It's promised. It says we're going to write a different paper about that, but I couldn't find any subsequent paper on that. So we don't know some of the outcome from some of the tests. In terms of IQ, they do tell us about IQ and the Tower of London test. The groups weren't matched for IQ. So already you've got that IQ difference. But they did find that the patients who'd had puberty blockers did poorly on the test of executive function, the Tower of London test, even when they controlled for that IQ difference test. So again, kind of what we might think, it's a frontal lobe test. And if you suppress puberty, you're not doing quite so well. Interestingly, the results of that are subsequently reported as saying that there's no detrimental effects on cognitive function. So it's you always need to go back to the original studies. I think that's your kind of take-home message there. And one of those was actually in the NHS policy document, that it was cited as saying that there was no thing. But this is kind of, this is a single case study. So we can tell very little from a single case study, very little. And this was an 11-year-old male to female patient who did have a baseline assessment. They were assessed at 11 years, they were assessed at 13 years, and then they were assessed at 14 years. Over the course of the treatment, they lost 15 points in verbal comprehension, 10 points in global IQ, and 9 points in working memory. It's only a single case study, but again, the results are completely in line with what you would expect. So because this is a standardized test, it's stable. So if you start off with an IQ of 100, you should have an IQ of 100. So that it's always standardized. So you would expect a straight line. Okay, and this is a really interesting study. So this study, they started off with 119 people, and then they looked at 72 of those. They sort of with IQ. So they did a really good baseline. They got an IQ on everybody, and then of those, they had a 40% dropout rate, but then they had sort of 72 people in their follow-up. But instead of doing another IQ test, which would really tell you what had happened to these people time 1, time 2, what they did was just dichotomize them and said half of them were vocationally educated and half of them weren't, or half of them got a kind of higher higher vocational education. So they just dichotomise them. So what this study tells us is that before treatment, IQ was normal. 30% of people who take puberty blockers and cross-sex hormones who undergo surgery, because they had all of those things, are educated to a higher vocational level after all of that. It doesn't tell us anything about the impact of puberty blockers on IQ. It's got a really high dropout rate. It's completely uncontrolled. And the simplified proxy measure of outcome is just extraordinary. It's like looking at, if you were going to look at the impact of puberty blockers on height and you measured everybody, and then your outcome is, can they get on a fairground ride, yes or no? I mean, it's that kind of proxy measure, so it's not particularly helpful. So, in terms of conclusions, I didn't find the kind of Nobel Prize winning, guess what, actually you can pause a developmental stage and then restart it and everything will be okay. I didn't find anything because there's just no evidence out there. And I think that's the kind of key take home from this is how has this been allowed to develop as a treatment when we know so much about brain development and stopping puberty and what happens in puberty. And we can kind of predict that cognition is going to be really, really important. How has nobody been looking at this? All of the thousands of patients that have gone through this treatment, I think it's a medical scandal. That's my particular point of view. So just to conclude, puberty is a sensitive window of neurodevelopment associated with significant cognitive changes in brain function and structure. Animal studies indicate that the suppression of puberty impacts brain structure and the development of social and cognitive functions in mammals. The effects are quite complex, they're sex-specific. There's no evidence that cognitive effects are fully reversible. No human studies have systematically explored these treatments on neuropsychological function with an adequate sample size, baseline or follow-up. There's some evidence of a detrimental impact in the very, very poorly controlled and very, very few studies that are out there. And really very critical questions remain unanswered about the nature, the extent, the permanence of any arrested development of cognitive function that may be associated with these treatments. If you're going to pharmacologically stop puberty, we need to know what is happening. Now, you are people who work in these settings. How many of you have got a neuropsychologist? Because you need to have a neuropsychologist. You need to have a neuropsychologist assessing them beforehand, and you need to have them assessing the outcome afterwards. So neuropsychologists really should be an integral member of the multidisciplinary team caring for people, and you need to be monitoring these impacts.